Thank you to the organizers for the honor to speak here today. And thanks to my co-authors for making this research fun and rewarding. Today, I want to talk about contact networks and pathogen transmission in wild animal populations. One of the conceptual challenges at the interface of animal behavior and parasitism is to better understand transmission. Successful transmission of a pathogen depends on contact between a susceptible and infected host, and the probability of transmission given contact. Put another way, the probability of a contact, often driven by animal behavior, multiplied by the conditional probability of transmission given contact, often based on host physiology and pathogen characteristics, can lead to a successful transmission event. And then, of course, we can have infection-induced behavioral changes. For free-ranging wildlife, it is often more practical to quantify contacts between individuals rather than successful transmission events. These contacts can act as a proxy for transmission by assuming that a fraction of contacts result in transmission events. The contacts between individuals in a population can be visualized as a contact network. Here, nodes can represent individuals or groups of individuals, such as a pride of lions. And edges between the nodes can represent contacts between those individuals. We find that indeed, most animals have a heterogeneous contact structure, where some animals have more contacts than others. For example, some lion prides have more contacts than others. Similarly, some raccoons have more contacts than others. Contact network studies have been used to explore mechanisms of parasite transmission in wildlife and have been utilized to ask some cool questions such as, are there trait-based features that predict super spreaders? What factors mediate individual variability in susceptibility and exposure? How does group living affect the spread of parasites? And are there feedbacks between network position and infection status? There are other ways that contact network topology can help inform understanding of transmission and disease dynamics. For example, we can take a contact network, simulate stochastic infection processes on it many times, and produce various transmission networks. These transmission networks are often the output of interest. With transmission networks, you can ask lots of cool questions. For example, when African lions were hit with a fatal outbreak of canine distemper virus, we asked if the observed canine distemper virus outbreak could be explained by lion-to-lion behavior and transmission. Or were other carnivores likely involved in spreading the virus? Were nomadic lions super spreaders? For raccoon rabies, we tested for seasonal differences in raccoon contact behavior. And we asked what implications might changes in contact behavior mean for the spread of rabies in urban environments. We asked, are current oral vaccination target levels enough to prevent rabies outbreaks in urban areas? For protecting the endangered Hawaiian monk seals from distemper viruses, how should managers prioritize targeted vaccination? We asked if targeting the most social seals was both efficient and effective. Finally, for understanding disease spread in the solitary Florida panther, check out Marie Gilbertson's poster this afternoon. So while we can do lots of cool things with networks, getting a contact network which is relevant for pathogen transmission can be challenging. We need to be able to define a pathogen-relevant contact, make informed choices about the method of data collection, and make informed choices about the analysis of the contact data. We can then use the contact network topology for transmission inference and better understanding of disease dynamics. Let me take you through each of these steps in gray right now. First, how do we define a pathogen-relevant contact? Here, we need to identify the pathogen of interest. We need to identify the focal host species and define the type of contact needed for pathogen transmission. For example, the type of contact needed for a pathogen transmitted by, say, fecal-oral environmental contamination is different than the type of contact needed for transmission of a sexually transmitted disease or infection. Another way to think about it could be thinking of contacts on a continuum of spatial overlap of home ranges, as would be relevant for indirectly transmitted pathogens, to two individuals needing to be in the same place at the exact same time as would be for a directly transmitted pathogen. Once you have figured out what type of contacts you need to capture, the next step is to choose the best method of data collection. There are direct techniques to collect contact data, such as behavioral observations. There are also indirect techniques. For example, you can use radio tracking data, for example, very high frequency or global positioning system collars to infer contacts. You can use proximity data loggers. You can use capture, mark, recapture, for example, trapping networks of rodents. And you can use RFID readers and PIT tag readers and other things as well. Now, one hopes that the different methods of data collection describe roughly the same contact behavior. However, different networks can and do emerge. For example, Sarah Perkins and co-authors found that contact networks derived by home range spatial overlap, as determined by VHF radio tracking, differed substantially than those networks derived from capture, mark, recapture on a trapping grid. The third step is to choose the best method of analysis to extract the contacts from those data. So for example, Marie Gilbertson conducted movement simulations where she knew the exact locations of all the individuals in her population and exactly which individuals had contacted each other. She made a contact network with the true contacts. She then changed her contact definition to mimic two types of common analyses. For the first, she loosened the window of time and the distance with which we called a contact. So we got more inclusive definition of contact. With the second analysis, she calculated the home ranges and looked for spatial overlap between individuals. You can see that the contact networks are different and that, again, we have to be careful in the decisions we make. So in summary, while network approaches continue to help us answer questions related to animal behavior and parasitism, we need to think carefully about the definition of a contact, the method of data collection, and the analysis of the contact data in order to get the contact network topology most representative of contacts relevant to the pathogen of interest. The process of constructing contact networks must be rooted in an understanding of the biology of the host parasite system and align with the relevant mode of parasite transmission and the host interactions necessary to facilitate it. Thank you for your attention today, and I'd be happy to take any questions later on live. Thank you.